Welcome to Chapter 3. In this chapter, we're really going to be discussing the structure and function of cells. And so we'll go through some of the different cell parts. We'll look at what a cell is, how they're organized, the plasma membrane, and how substances cross over it. Then we'll look at the parts of the cells, including the nucleus and the intramembrane system, talk a little bit about protein structure and function and how that's made, the cytoskeleton, cell movement, how we join cells together, and the metabolism and the energy reactions. In this first portion, what we're going to do is talk about just the cell itself. So we want to understand the basic principles of cell theory. We'll look a little bit at why cells are so tiny, and then we'll look at some of the tools to study these cells. Well, cell theory, as we've learned in the last chapter, is that the cell is the basic unit of life. All living things are made up of cells, and new cells always arise from pre-existing cells. So then we have to ask, why are cells so small? Well, we have to think about the surface area to volume ratio. Little tiny cells have a larger surface area compared to the volume inside of it. More surface area means nutrients can pass into the cell much faster and get to the center much quicker, and wastes can leave the cell. If you get it really, really large, that surface area becomes small compared to the volume, and then it becomes quite difficult to get the nutrients into the cell, and it will actually die. Here's an example of that. You can see if we took a single cube that's 4 centimeters by 4 centimeters by 4 centimeters, what you will find is the total surface area changes depending on how much we break it apart. But the volume actually remains the same. So the amount of material that's present doesn't change, but the amount of surface that things can pass in and out of does change. So you can see in the first cubed example, we have a 1.5 to 1 ratio. Just cutting it up into 8 smaller cubes changes it to a 3 to 1 ratio. But then if we get really small pieces, we get a 6 to 1 ratio. And it's when we start getting into these tiny little pieces that we start understanding why it's so important for a cell to be small so that we can efficiently get nutrients in and wastes out of the cell. Now, if we want to study the cell, we'll find that there's some different types of microscopes that we can use. In the classroom, we tend to use a compound light microscope. It has a lower magnification and just simply shines a beam of light through the specimen. But what's cool about this is you can view live specimens. Now, there's a limit to how much we can magnify with a compound microscope, so then you can move to a transmission electron microscope. It's also going to have a two-dimensional image, and it's using electrons to view these internal structures. It's got a very high magnification, but we have to kill the specimen in order to be able to mount it. We can also use a scanning electron microscope, which does make a three-dimensional image. It's also using electrons, but it's using it to view the surface of those structures. Again, high magnification, but no living specimens. Here you can see a comparison of looking at red blood cells in these 3 different models. Here is just a regular compound microscope. We've magnified about 250 times normal size. We can see those red blood cells. Here's a transmission electron micrograph. Again, it's a two-dimensional image. It's about 10,000 times normal size, so you can really start seeing some things here. But the scanning electron microscope is the one that really gives those beautiful three-dimensional structures, and we could get much closer than even this. So now let's start talking about how we have these cells organized. We're going to try to identify the components of the human cells and the functions of each of those components, distinguish between the structure of a prokaryotic and a eukaryotic cell, and then summarize how eukaryotic cells evolved from prokaryotic cells. So first, I want you to think about prokaryotic versus eukaryotic cells. In the past, when we've talked about them, prokaryotic cells are itty-bitty, teeny tiny. These are things like bacteria. They are so itty-bitty, they don't even have a nucleus. They just have a little loop of DNA. It's likely that these were the first cells on the planet. Eukaryotic cells are much, much larger. They have a nucleus that houses DNA. They have membrane organelles. In fact, a bacterial cell is about the same size as a mitochondrion inside a eukaryotic cell, so much, much bigger than prokaryotes. We can tell you that there are some things that they all have in common, regardless of being prokaryotic or eukaryotic. They all have a plasma membrane, cytoplasm, which is that kind of goop that's inside the cells, and then DNA. But beyond that, we can start looking at the eukaryotic cell itself and notice that it has a tremendous number of individual organelles. Remember, those prokaryotic cells are probably similar in size to this mitochondrion right here. Look at the degree of structure that we have in the eukaryotic cell. If we start by just looking at the nucleus and the endomembrane system, what you'll find is that these structures are all involved in protein production. The nucleus is containing that genetic code on how to assemble a protein. The ribosomes actually assemble the protein using that code, and the endomembrane system, or your endoplasmic reticulum, will help produce that, and then the Golgi apparatus will then package it. So the nucleus itself is surrounded by a nuclear envelope. We have chromatin, which is the DNA when it's unraveled. It's a chromosome when it's raveled up. And a nucleolus, which is a clump of both DNA, RNA, and it's sort of responsible for making the ribosomes. So here you can see that nucleus. There's the nucleolus right there, the nuclear pores. And then you see the endoplasmic reticulum out here that's just littered with those ribosomes. This is where most of the protein manufacture is going to occur. Again, the ribosomes are these little tiny organelles made of rRNA and protein. They typically are found in the endoplasmic reticulum, but you can see them around in the cytoplasm as well. And this is what manufactures the protein. In the endomembrane system, we're seeing a series of membranes where these molecules are transported through. It consists of the nuclear envelope where the little messenger RNAs come out, the endoplasmic reticulum where we can see the manufacture of these things formed, the Golgi apparatus where we see packaging occur, and then we can also have these small little packages of, in lysosomes, digestive enzymes, and vesicles, just some storage. Here gives you a better picture of it. Our nucleus with the code for how to make proteins. A copy of the code is sent out into the ribosomes along the rough ER. It then encounters the Golgi apparatus where we can have packaging. We can have things like secretory vesicles that are then secreted outward. We can package things like lipids and such in the smooth ER as well. The rough endoplasmic reticulum is studded with ribosomes that are used to make your proteins. Smooth ER lacks ribosomes, but it aids in making carbohydrates and lipids. And then again, we have the Golgi apparatus, which are these flattened stacks that package and process the proteins and lipids. Lysosomes and vesicles I mentioned back on the last picture. These are the membrane-bound vesicles that are made by the Golgi, and they contain digestive enzymes. These are kind of your trash cans of the cell. It's where you digest parts that you don't want. And then the vesicles, which are small membrane-bound sac that are used for transport. Another structure in the cell is the cytoskeleton, cell movement, and cell junctions. And we're going to talk a little bit about what those parts are, how they're made, and what they do. The cytoskeleton is a series of proteins that maintain cell shape as well as anchor and move organelles around inside the cell. They're typically made of these proteins like microtubules, actin filaments. There's even some medium-sized intermediate filaments, but these are all used to kind of give the skeleton inside your cell structure. Cilia and flagella are also made of proteins, but they are put together in such a way that they allow movement to happen. Here you can see the shaft of a sperm. It has these microtubules that allow for the movement of that sperm. Here you're seeing cilia. This was probably taken out of a lung where you can see these little tiny hairs that, well, they're not really hairs, but they're made out of proteins that allow you to move things around in your lungs. Cell junctions also are made of proteins, and we can use 3 different types of junctions in order to put cells together. Adhesion junctions, you can see the proteins actually physically lock the cells together. You use these in places like skin that need a really tough binding. Tight junctions are used where you don't want any leakage to occur. You can see these are sealed together really nicely using these very tight protein junctions. It's almost like a zipper-like barrier. Again, we use it where you don't want leaking to occur. Places like the kidney, the digestive system. It's not necessarily going to have a lot of physical stress, but you want to have a nice tight seal. The last one are the gap junctions, and you use these where you want to allow for things to be able to move back and forth between cells. So they actually have these little tiny holes within them which allow channels. Places you might want this is like in the heart, where you want those molecules to transmit very quickly so you can get that coordinated movement. So again, we went very quickly through some of the primary structures within the cell. You can see there's the nucleus with the DNA, which codes for protein. There's that nucleolus. Pores, where messenger RNA contain small bits of the codes for how to manufacture proteins. The endoplasmic reticulum littered with ribosomes for protein manufacture. Smooth ER, here's the rough ER, here's the smooth ER for manufacturing carbohydrates and lipids. The things that are manufactured head over to the Golgi apparatus where they can be packaged. There are things like lysosomes where we see digestion of things that we don't want can occur. Again, those are manufactured by the Golgi apparatus. We also have vesicles of those smaller proteins. And then littered throughout, you can see these structural filaments in here that can be used for the cytoskeleton. They can be organized in ways that movement can occur. And then you can also use those to glue cells together in 3 different ways, depending on the different functions of the cells. All right, that sums up this section. Thanks, guys.